Probably most of you heard about genome-wide association studies, but maybe not all of you. So let me just quickly, right? In these studies, you take a lot of patients, you need a large number, and a large number of healthy people, and you look at their sequence, and you look for differences in mutations that are associated, that you find them that are more common in the people in one group than in the other, okay? And these are very remarkable kind of endeavors. It's the technology and the coordination involved. And there's been quite a few of these GWAS, is the shorthand GWAS studies. And you find all kinds of associations. You find all kinds of mutations. Actually, you find too many of them because the genome is a large place, and you find lots of things just by mistake that are very common in one group than the other. So it also requires a lot of statistical sophistication to really tease out mutations or associations that you trust. And people have done that. And they've come up with a set, if you combine all the important GWAS studies, recent years, there are several thousand of these mutations. Each of them, or for each disease or condition, there are several mutations, maybe hundreds of mutations associated with that. But there is a mystery, because the majority of these mutations, of these GWAS SNPs, they're called, we don't know what they do. And if you don't know what they do, if you can't associate some mechanism to that mutation that you find, it's hard to kind of act on it and to get to the point where you want to get to, which is some drug that will target something specific and will make life better for someone. So the talk today, in part at least, is about how ChIP-Seq and epigenomics data that we're talking about here helps with that. So, and it's really understanding some function of some places on the genome. So I'm going to talk or mention work of many people, the BroadE Epigenomics program, the production group specifically, and Brad Bernstein's lab, and Manolis Kellis, and mention data from ENCODE and approaches that related to data collected for ENCODE. So, right, this is a big question in biology. What does the genome do? That's what we want to know. So what do we start with? I mean, it's not exactly start with. Things have started long before sequence. But we do have the full sequence now, right? And when you look at the sequence of the genome, you can find some patterns just from that information of those letters. You can find that there are special places that something is going on. And not going into too many details, or not going anywhere, okay, you can actually identify genes, places where proteins are being transcribed and then translated, and we understand genes more or less. I mean, it's not that every protein we know what it does, but we can find those places in the genome where there are genes. But if you now look at what I talked about before, those genome-wide association studies and the SNPs that come out of them, these mutations that are associated with some condition, some of them, can you see this pointer at all? Probably not. And those of you who are looking at that screen can, yeah. Those on the gene, that's something we can understand. There's a mutation. It may have changed the structure of some protein and it cannot function anymore. But a lot of those mutations fall somewhere that we don't really know what it's doing. It's that with a question mark. Because most of the genome, we don't really know what it's doing. So let's leave that aside and talk a little bit about epigenomics. Chuck already touched upon the notion. No, I'm sorry. I forgot to say a couple things that I wanted. I'm going to have this kind of pretty schematic talk about things like what you just heard me talk about. One comment from last year was that people were expecting some level of discussion about actual data analysis. Alon is going to do some of that in the next part. He's going to talk and actually help you actually do some alignment and some of the basic processing and some of kind of rudimentary analysis of ChIP-Seq data. But if at any point during this talk or after it, you have questions about data analysis of this kind of data, I'd be happy to try and answer that. And I'll try to make this part of my presentation, basically the presentation, I'll try to make it not too long, to leave enough room for that. Okay, with that. Chuck already mentioned this epigenetic part of this world, right? The idea is that a liver cell, when it divides, it doesn't suddenly become a muscle cell. So it has some information that's telling it it's a liver cell, even though the liver cell and the muscle cell in my body have exactly the same DNA. So that information is not in the sequence, it's somewhere else. There is a different level of information and regulation, and that's epigenetics. A very promising candidate for the physical carriers of that information is what we call epigenomics. That's everything that resides physically on the genome and kind of interacts with it in different places and does something to it, except for the DNA sequence itself. So in epigenomics, we talk about DNA methylation, for example. We talk about histone modifications. That's mostly what I'm going to talk about, or the kind of data I'm going to mention. That's because that's the focus of our group. That's what Chuck explained a little bit about. I won't go into the details, but summarizing what some of what Chuck said, DNA is wound around these beads, the nucleosomes, which can be chemically modified. They have these tails that can be chemically modified. And when they are, certain modifications can, for example, make the DNA wrap a little less tightly around the nucleosome and kind of open it up for transcription, so it can activate genes. And in other places, another kind of modification or set of modifications can cause the DNA to be more tightly kind of wound and closed and stop it from, kind of stop any activity there. So that's the kind of things that histone modifications may be doing. The kind of data we're talking about is ChIP-Seq. Here I'll give you the cartoonish kind of, I come from physics. This is a physicist view in a way of you heard the biologist view. This is a physicist view of ChIP-Seq. So these things here, I mean, this is the genome, right? That's a very realistic picture of the genome. With some target, something on the genome that we want to measure where it is, that may be a histone modification, right? We want to know where, right? There are these histones all along the genome, and some of them have a certain modification, and we want to know where those locations are. We want to create a map like that. So we break of course, we first cross-link or digest, but we chop up the genome into small pieces. We then add antibodies that are mostly, they prefer to go to the target that we want, but sometimes they go elsewhere. Then we can wash away everything else, but we pool down the pieces that we want. Now we don't need anything anymore. We don't need the antibodies. We don't need the target. All we are interested in are these little bits of DNA because we can align them. And once we align them, we can, we can sequence them. We sequence them so we know what their sequence is, and we can align them. We can find, for most of them, where in the reference genome they're from, and we can pile them up like this and create maps that look like this and like some of the figures that Chuck showed you. Okay, so this is an example of some chromatin maps. And they tell you in this location, there is enrichment for this histone modification on the genome. Okay? That's what these maps tell you. And ENCODE, part of ENCODE was to do a lot of these for a lot of cell types. So, yes, this is what it looks like. So it was, a bunch of histone modifications. This is not a histone modification, CTCF, but it doesn't matter. And lots of cell lines and cell types, and we are now in a new phase of ENCODE. And data looks like that, a lot of tracks. And what I really want you to see here in this complete PowerPoint mock-up of tracks, but it doesn't matter, is that there are all non-trivial molecular events going on everywhere in the genome, not just where the genes are, but you can see there are lots of places that something is happening. Something that is completely non-random and is coordinated sometimes across different histone-modifications in the same cell type. So there is a lot more now that we can say something additional about. So one of the first things that were done with this data is to start saying, okay, there are many flavors, maybe many types of histone modifications. Are there any connections between them, or do they just occur in any combination whatsoever? Like, are they completely independent, each of them doing its own thing? Or do they tend to do things in some relation to one another? And it turned out that there are a very small, if you can consider something like 10 or 12 histone modifications, there are many, many combinations that you could see them on the genome at many thousands. And it turns out between 10 and 30, depending on how sensitive you are, or strict you are in your threshold, there are only about maybe 10, maybe 15 combinations that really show up a lot everywhere, combinations of histone modifications. And if you look a little further into where these combinations occur, they occur in places that make sense, and some of them reveal new sense. So, for example, you find them at promoters, but then you find that there is one combination that appears at the promoter of a gene that's active, and a different kind that appears at the promoter of a gene that's not active. And there is something that really marks the body, the part of the genome that's being actively transcribed. And of great interest is this set, which is not the only set of things away from genes, but it's an important one, the enhancer set, which are regions in the genome that are far away from genes, at least in this linear look, but act as remote switches. They actually activate genes if something happens at the enhancer. So there are a handful of them that were known before that allowed, and there are different factors that are known to be involved with enhancers that allowed the inference that a certain combination of histone modifications is probably associated with enhancers, and then you find them in many, many places that were not known before. So you have this new, you're now armed with a lot more annotation and information about what these different places in the genome do. So what do you do with this information? So this was kind of laid out in this paper there. A roadmap for annotating the desert was not the title of the paper. I can't remember the title of the paper. This is what I think is the important thing that came out of it. And there has been several works that kind of are following up on this. This is by no means the only way to use epigenomics and kind of ChIP-Seq data to learn about biology. And I can talk about a few others, but I find this is one of the most exciting things that came out of ENCODE. So first thing, in each cell type, right, for a particular cell type, you have these different tracks that show you where different histone modifications are. And you have already identified what combinations correspond to different types of functional elements that you're interested in. So you can identify in a particular cell type, oh, here is a set of active enhancers. Okay, you do that, but you have several cell lines. So you do it for each of the cell lines. And you actually combine, you kind of create this master list of enhancers. Mind you, most of these enhancers, you don't really know which genes they're acting on. There are different ways of trying to make that associations. Sometimes it's the kind of nearest, but nearest can be 50,000 base pairs away. And you compile this master list of enhancers by cell type, and you say, okay, this enhancer is on in this cell type, but off in these other cell types. And this is on here and off there. Okay? So here's an example. This is kind of a illustration on something which is a very early prototype of a tool that we'll see better days. But it doesn't really matter right now. What you have here is basically a graphical representation of the matrix I just talked about. So each column is No, I should, let me take it, let me think just 1 second. Each column here represents a GWAS SNP, okay? So remember we talked about these mutations? So I now look at all the regions in the genome, all those places where there is a mutation associated with a disease, okay? And then I limit my view to only those places where there is an enhancer found from our data, okay? So that's the two top rows. The first one shows, I mean, the details don't matter, but each kind of shows a SNP that came in a GWAS. The second shows you there is a candidate enhancer there. That means there is an enhancer that is active in some cell type. That's it. That's what you see here. And what you see below are the different cell types, and for each of them, which of these enhancers are on in that cell type, okay? And so this is a very crude kind of, I wouldn't even call it analysis. This was like a very quick exploration, but I think it illustrates the idea. If you look here, these are, this is a brain cell type, hippocampus middle. And you see there is a cluster here of enhancers that are on only in the brain. And when you ask up there, okay, so what are the SNPs that they are found in the vicinity of or overlapping these enhancers, you find that most of the SNPs up there in that cluster are associated with Alzheimer's. So that makes a lot of sense. You have identified some mutation that was associated with Alzheimer's. We didn't know what it was doing because it was away from genes. But now we have better understanding. We can say, okay, it's actually in an enhancer. And this enhancer is active only in brain cells. So it's probably doing something in brain cells. So it makes sense. And you can continue more after that, right? You can take all the enhancers that are on together in a particular cell type and look for sequence motifs there. Because the thought is to turn on an enhancer, the histone modifications need to kind of, I mean, and I'm not even sure that the causal relation is really known, but they have a certain typical chromatin landscape in the sense of histone modification landscape. But in addition, you need an actual factor to bind at the enhancer and do something to the gene. So when you collect all the enhancers that are kind of active in the same cell type and look for sequence motifs under them, you find motifs that can tell you, okay, these are known binding motifs for some transcription factor, and you can go and check, oh, that transcription factor is actually on exactly in the same cell type or more in the cell type that these enhancers are active. So you can start making a pretty detailed picture of what's going on. Right, so we added a lot of information now with these kind of data to those unknown parts of the genome. We can say about that element, okay, it's on in particular cell types and not in other. We can connect it kind of by finding the motifs. We can see what's upstream of it, what are the transcription factors that are activating it. There are different ways of doing correlations and other methods to try to figure out what is the gene that this enhancer is acting on. And here are a couple of examples where this can lead you. That's from the same paper. So in these two examples, a mutation, a GWAS SNP was found. So it's a mutation associated. I mean, it's not my exploratory little thing that I showed you before about the Alzheimer's. No one ever followed up on that. That was really a quick and dirty thing. This is the real thing where the investigator that went after a particular mutation that's known to be associated. These are two diseases, two cell types. I don't want to go into the biology because I know I'm going to trip there. And in each case, the mutation hit modified a known motif that was part of the enhancer. And actually in these cases, it made the motif stronger. So probably the enhancer got stronger, or maybe the gene was overactivated. So there is a mechanistic kind of map here that can lead to the next step of, okay, maybe we can use that. In fact, in a more recent detailed study focusing on immune diseases, a slightly different pattern was found, that a lot of mutations do indeed hit enhancers, but away from the known motif. They're still associated highly with the disease, and they hit an enhancer that is specific to the immune cell types, but it's not at the motif, so they're changing the function of the enhancer probably in some other way. That's it for this part. I'd be happy to take questions if you want, either related to this or take me in completely different directions. Yes? The enhancer track is, I mean, yes, you can get it from ENCODE. They compiled, so this assigning a chromatin state to a particular part of the genome based on all the histone maps was done systematically on all the ENCODE data using a algorithm and a program called Chromatin Maps. And you can actually read it off of there. But there are other ways that people are calling enhancers now, slightly different methods. I mean, the very last thing about the immune stuff that's going to be published very soon, they have a different way of calling the enhancers. But yes, you can use ENCODE data to find this.
Also, do enhancers or
Enhancers are not, you said if enhancers are transcription factors? No, so enhancers are parts of the genome, particular locations on the genome that are the location of a switch. So if something else happens there, they turn on a gene. The thing that would happen there would be the binding of some molecule. And a molecule that binds to the DNA and regulates expression, that's a transcription factor. So transcription factors often would bind to enhancers. Yes?
Can you take into account something like allelic fractions or cell type heterogeneity, for example? Like,
I'm sorry?
Is there any way to do some type of allelic fraction estimation? Like, for example, like we did that in whole exome sequencing, or like some way to take into account like any heterogeneity within your sample?
Heterogeneity within the sample.
Probably not right, because when you just pull it down, you don't get the other. You only get what you grab onto, right?
Right. So allelic variation, you can, because you get the sequence, and you can find. I mean, it depends on the depth of the sequencing and how much, but if you have lots of information from a lot of assays done on the same cell type, especially if you have good sequencing of the, parental sequences as well, like in some of these cell lines, you can actually say this, and you're lucky, then you can say about certain reads, this fragment actually came from this allele, this came from that allele. And people have done studies showing sometimes allelic asymmetry, like certain modification would only pile up on top of one allele and not the other. But you need more data, but these things are definitely happening. In terms of heterogeneity within the sample, there's a whole new, to really do something like that, you need to collect information about single cells. And I've actually been part of a collaboration between a physics lab here at Harvard and a chromatin biologist from Brad Bernstein's lab to use nanodrops to put cells in drops and then do ChIP-Seq there. And we have data, and we're seeing some things. It's a tough technology and hard to, but we're trying to make sense of these data.